molecular oncologist, which means I'm interested in cancer, how cancer grows and how we can stop it growing and how we can kill it. But I'm interested in it from a molecular point of view. So we look at the DNA inside the cancer and we find out how normally the DNA is replicating, how it's growing, how it's being repaired, in order that we can target these pathways to develop better cancer drugs. How our standard cancer drugs work. They inflict lots of DNA damage so that any rapidly growing cell dies. Historically, these have worked quite well. However, resistance can arise in cancers, and generally this comes about because our own DNA repair systems are too good. And when these are upregulated in cancer, the cancer drug can repair all the damage and therefore the drug doesn't work. So historically, animal models have been used to recapitulate the tumour and how it would be inside a person. However, I believe these aren't particularly clinically relevant because ultimately a person and a mouse are very different. So what we want to do is to, instead of grow tumour cells on a flat plastic surface, we want to grow little mini tumours in the lab and see if we can target these with drugs. We found some really cool stuff as part of our THRT funded project. We found a totally new role for a protein, UPF1, which seems to be really important in cancer drug resistance. We found a totally new role for this protein in DNA replication and repair, and we want to further study this to see the implications of this in cancer. We know there is an implication because we can see from the data that exists that when tumours have very high levels of UPF1 or very low levels of UPF1, they respond very differently to cancer drugs, and this correlates with very poor patient survival. So we need to find out what's going on in cancers with UPF1 and how we can treat these cancers.